Hi, welcome to the 6th week of Consider This, Simplifying the COVID-19 Conversation. This week we'll be hosting Dr. Kyle Bibby, who will talk to us about detecting the virus in wastewater. So many of you may have been hearing about this term PCR, which stands for Polymerase Chain Reaction. So most of the time we think of it in terms of diagnosing the virus if you're infected with the virus, but we can also use this technique to isolate or detect the virus in environmental samples like wastewater. So just to remind you of some terminology, we go from DNA generally to RNA to protein, the building blocks of a cell. And actually PCR usually recognizes DNA, and it goes through cycles of temperature gradients, and one cycle will duplicate the DNA. So we go from 1 copy to 2 copies in cycle 1, to 4 in cycle 2, and to 8 in cycle 3. Now, if you remember, the SARS-CoV-2 virus is an RNA virus, and so that complicates it, that we can't use the standard PCR to detect it. And so we have to use a specialized technique called RTPCR. Now, the RT stands for reverse transcriptase. And so essentially what that does is it reads the RNA and changes it back into DNA. So we isolate the virus and the RNA from the virus. We add in the enzyme or the protein that reads the RNA, reverse transcriptase, and it really makes just a copy of that RNA into DNA or cDNA. And the C doesn't stand for copy, unfortunately, it stands for complement DNA. And then we amplify the cDNA in our subsequent cycles, amplifying it and detecting it. And you might also see a term called RT-qPCR, and the Q stands for quantitative, in that we can just actually measure the amount of cDNA in a sample. So in general, when we do the reverse transcription, we copy the RNA genome of the virus. And if you remember, there were different parts of the genome that encode specific parts of the protein, like the spike protein or the nucleocapsid protein, etc. And in general, we're only amplifying a certain part of that genome, and we call that the target sequence. And so what we do is we put a primer that complements a specific region that we want to amplify, and we anneal it to the cDNA, and that we do that at a certain temperature. Then we put in the polymerase, which reads the cDNA and makes a complement strand. And we call this the extension phase, and it's generally at a little higher temperature. And then we separate these two strands or denature them at yet a higher temperature. And this is called a cycle. And we have subsequent cycles of annealing, extension, denature, each time amplifying the template even more. And in general, when we do RT-PCR, we do about 40 cycles. So let's see what it looks like. When we are binding the cDNA, so the cDNA has a certain sequence of nucleotides, A, G, T, T, we put in the primer that complements that. So A's always bind to T's and G's always complement with C's. So we put this in and we know we want to amplify this certain region. Then the polymerase reads those different nucleotides, adding the complement nucleotides to the complement strand. So in cycle 2, we again add our primer, but now we have this green strand or this complement strand, which is actually a different sequence, so we have to add a different primer. We add the polymerase, and it reads and copies or makes the complement strands to each of those. We denature, and we go to cycle 3, and we do the process again, reading, making the complement strands, and denaturing. And so by cycle 4, we actually have 8 strands. We went from 1 to 8. So how do we actually measure these strands or these DNA? So the way that works is you have your primer, but you also put another primer or complementary strand called a probe. And that probe is connected to a fluorescent molecule that emits light. But the probe also has a molecule attached to it called a quencher. And so when the quencher is in close proximity to the fluorescent molecule, you get no fluorescence. The polymerase, as it reads the cDNA, degrades that probe and separates the quencher from the fluorescent molecule, so you get an emission of light, essentially. And you can imagine the more strands you have, the more complements you make, and the more fluorescences that you get. And we're able to measure that fluorescence each time it goes through a cycle. And so you can see that in this graph here. On the y-axis, we have fluorescence. On the x-axis, we have cycle number. And there's a certain threshold of a background noise, essentially, that even if you didn't have any virus, you would still get some fluorescence. And so what we do is we measure the cycle number in which the fluorescence crosses that background or that threshold. And this is called the cycle threshold, or the CT value. In this example, in the green example, it crosses the threshold at CT20. You can imagine you have another sample that crosses a little bit later, say at CT21. And this is only a difference of a single cycle. And if you remember, each cycle, you double the template. And so you can assume that the starting amount in the green sample was about twice the amount of the cycle in the blue sample. And you can have other CT values that come up much later, so at cycle 35, and some that never even cross the threshold. And it's generally thought that past cycle 35, we're just measuring perhaps residual RNA in the sample and not intact infectious virus. And so generally speaking, if the CT value is less than 35, we call that a positive sample. And if it's higher than CT 35, we call that a negative sample. I'm not exactly sure what happens in wastewater testing, but I'm sure we'll hear on Monday night. So I hope you join us, and I'm looking forward to it. Thank you.